The kidneys are complex organs that keep our blood clean and balanced, making sure that we stay alive. During kidney failure, your body fills up with waste and fluids, which is both painful and very deadly. Kidney failure has no cure, so these patients will rely on dialysis or transplants just to stay alive. Now imagine having to go through all of this as a child. 1% of our population is born with a disease called congenital anomalies of the kidney and urinary tract, abbreviated as CACUT. This can cause kidney failure in young patients. CACUT is caused by the disruption of kidney development. The cells are not ending up where they need to be. This can result in the failure to form kidneys or otherwise forming malfunctioning kidneys. During normal kidney development, the cells not only need to divide, but they migrate to where they need to be to function. So what is different about the cells in CACUT kidneys? I began answering this question by generating a list of genes that have altered expression in a mouse CACUT kidney model. A subset of these genes encode proteins that are known to be involved in a specific type of chemotaxis. Chemotaxis is the movement of a cell in response to a chemical signal. I like to think of chemotaxis and chemical signaling as airport signage for cells. If you've ever been lost in an airport before, you can find your way by looking at the signs. You can move towards the signs that point to the terminal that you're trying to get to, while you stay away from signs that say things like no entry. For chemical signals, they can be attractive or repulsive to a cell. And as with signs, a chemical signal can have different meanings depending on the context. So a cue that is attractive to a kidney cell may very well be repulsive to another cell type. Now imagine the airport signs were in a language that you didn't understand. You wouldn't know where to go anymore. Just as with signs, in the case of chemotaxis, a chemical signal can only be understood by a cell if it expresses the correct receptor on its surface. So if chemotaxis genes are altered in expression during CACUT, are CACUT kidney cells getting lost during cell migration? To answer this question, I will be working with mouse embryonic kidneys that are cultured in a dish. These kidneys are no longer inside of the animal, so I can observe their development in real time, and I can also alter their gene expression. This way, I can deduce what these genes are doing during kidney development by looking at how kidney development changes when I turn up or down the expression of these genes. They could be what guide kidney cells towards them, or what push them away from where they shouldn't be. Or they could be what allows these cells to understand these signals. My research will broaden our understanding of what genes are instrumental to kidney development. This will help us understand why CACUT patients aren't forming their kidneys properly and open up potential treatment options for them. Thank you.